the one organization teaching. How much energy? Yes,
Hello, welcome, sir.
Hello.
Yes.
Good morning. Good afternoon.
Good afternoon, sir.
Actually, I just the session starts at 2:30, na?
Yes. 2:30 it starts, sir.
Yes. So I just want to just make sure that everything is okay. I will
Yes.
just share the slide. You please look at it.
So you are going to share from your end, right, sir?
Yes, that may be better for me.
Yes. Ye-
Yeah. Shall I share?
2:30 it starts, sir.
Yeah.
Yes.
So I will just share my slide. You just look at it whether everything is just smooth or not. Okay.
Okay, sir.
Yeah. Just give me 1 minute.
Okay.
Can you see my slide?
Yes, I'm seeing, sir. I'm seeing.
Okay. And I will just make it now. Can you see the full screen?
Yes, full screen I'm seeing, sir.
And is slides moving?
Okay, sir. Done. Thank
Perfect.
you. Thank
Yeah.
you very much.
So I will join them subsequently. Okay.
Yes, sure. Thank you, sir.
Thank you. Bye.
Yes, I'm seeing, sir. I'm
Sure,
seeing.
sure, sure.
Yes, full screen. Yes, sir.
Today, what I could see, the way whole panel discussion was all the speakers, panelists, they stick to time, come up with brilliant solutions. And the way our chair, from the conversation, really I could only remind the problem that the lens is so good. They were just sitting, raising your hand and they stop.
इस बार आप मोबाइल में देख लीजिए कर जाएंगे नहीं 11W कटा ठीक
Thank you very much. We had a wonderful session. I have quick notes today, so we are going to break the lunch. At 2:05, and we would have 6 conference sessions. Session 1 and session 2 is for
एक घंटे का ब्रेक दे रहे 205 और 305
thanks you guys for that.
305 205 बोल रहे हैं अभी 205 हो रहा है अभी हो गया
नहीं, as पर वो ये देख के बोल रहे होगा.
One is building small holders to trust farming system, security. Another special session is on public-private partnership for rice research and development. There are a lot of logins we'll be talking about. And then we have 2 poster sessions on one is for Team 2 on sustainable rice farming. Another is for Team 4, rice for livelihood security and profitability. Those who are presenting questions, we are requested to visit the Biotic clinic and answer any questions posted below to your designated. Just look at those and then come and join through the Zoom link to have an interaction with the co-chairs and coordinators for evaluation of the questions. So this is all from the organizers.
Save time for one to Mondal sir, namaste and welcome sir.
Now my question is, Anjani ji, you are ready?
Mondal sir, are you listening my voice sir?
Yes, sir. It's clear.
Yes sir. Good afternoon sir. Welcome sir.
Thank you.
Yes, yes. Sir, the recent information is that Dr. P.K. Chakravarti, so he will be joining the meeting sir.
Okay.
Okay.
Good afternoon, Dr. Rath.
Good afternoon, Dr. Mondal.
So everything is right. I think we can resume 2:30. Eh?
We'll start by 2:30, sir.
Sure.
So yes, both the co-chairs have not joined yet, sir. So once they join, so we can start, sir.
Okay.
Hello, Finbar.
Hello, yes?
Yes. Are you able to listen? Hello.
Yes?
Yes, that's nice.
Yes.
So we are going to share your slides. So once your turn comes, so you straight away start presenting. We are going to share your slides.
Okay, okay, that's great.
Will it be okay?
Yes, I thought I had control of the slides, but that's okay. So will I just tell you when to go to the next slide? Is that what you want me to do?
Otherwise, no issue. You can also share also, no problem.
Probably, will I just share them? Yes.
Yes, you can share from your end.
Oh, okay, that's great. Thank you.
Okay, okay. Thank you.
Thank you very much.
Thank you.
Hello, Dr. Kalyan Mandal.
Hi, yes, sir. Good afternoon, sir.
Are you able to hear me?
Yeah, sir.
Very good.
Yeah. So we are waiting for sir to join Dr. P.K. Chakraborty also,
Yeah.
so that we can start our session.
Yes, sir. Yes, sir. You are audible, sir.
Chakravarti is to join.
Yes, sir. He is about to join just 5 minutes. He has joined, I think.
He has joined.
Good afternoon, sir.
Namaskar, sir. Namaskar. My sir, namaskar.
Namaskar, namaskar. Rath has joined.
Dr. Vasana. Dr. Vasana.
Yes, sir. Yes, sir.
Are you hearing?
Yes, I am hearing, sir. I am hearing, sir.
I will
Hello,
start.
sir.
Now I will start.
1 minute. Just, sir, let me start with some instructions first, sir. So then we can start.
Okay.
Good afternoon, everyone. Respected chairs and respected co-chairs and conveners and all participants. So I am Dr. Basan Gowda. I am the technical host for this particular session. So I request all the participants to strictly follow the instructions sent to you regarding the conduct of this session. So once the session starts, so you need to follow all those instructions. So with this, I will hand over the session to Dr. P.C. Rath. Over to you, sir.
Good afternoon, everyone. A warm welcome to all of you to the technical session 2 on biotic stress management in rice. I am Dr. Prakash Chandra Rath, Head Crop Protection Division and a co-convener of this session. It is my privilege to introduce our co-chairs of this session. One of our co-chairs is Dr. C.D. Mai, President, Board of Directors, South Asia Biotechnology Center, New Delhi, and he was former chairman, Agricultural Scientist Recruitment Board. We also have Dr. P.K. Chakraborty, member, Agricultural Scientist Recruitment Board, as another co-chair. Dr. C.D. Mai and Dr. P.K. Chakraborty are very well-known name among the rice researchers of the country, and they do not need any further introductions. So now I would like to invite Dr. C.D. Mai and Dr. P.K. Chakraborty to chair this session. Over to you, sir.
Thank you, Dr. Rath. I think first of all, I will welcome my another co-chair, Dr. P.K. Chakravarti. Am I audible?
Yes, sir. Yes, sir.
Yes, sir. You are very much audible.
So basically, I also would like to welcome our rapporteur, P.C. Rath and Dr. K.K. Mandal. And we have so wonderful speakers also. I think this session we are going to devote 2 hours, nearly 2 hours, for almost 4 lectures. One will be a keynote lecture and 3 will be lead lecture. And then there will be some kind of a summarization. If you have any questions at the end. Let me first tell that I started my career with rice. And I'm very happy that when I see now the Cuttack Institute has started as a research assistant in 1967 with Dr. Padmanabhan, I find that the institute has grown now to quite a good extent. I also worked in rice in Punjab when I was there as a rice pathologist in the coordinated program. And I find that majority of those Biotic-stresses which existed then also are existing now in a different format. For example, if I say rice tumor was initially just was a minor disease, today it has become very significant part of India. For example, blast is continues to be there, and we have large number of chemicals like cyclazole and new chemicals. But then we have problem of export. So you take any field, there are how
सर सीडी में सर आपका साउंड थोड़ा फ्लक्चुएट कर रहे हैं. हेलो, आपका वॉइस क्लैरिटी थोड़ा सा फ्लक्चुएट कर रहे हैं. आई थिंक हाँ फ्लक्चुएशन हो रहा है वॉइस.
अब हो रहा है, एक मिनट. साउंड नहीं आ रहा बराबर.
Sir, your network is poor, sir. It is showing your network is poor.
अब लग रहा है नेटवर्क का क्या प्रॉब्लम है?
Sir, maybe you can switch up your video, then sound may become better.
Okay.
हाँ तो
Sir, now you can speak, sir. I have
Now, are you able to hear? Yeah.
Yeah, we are hearing, sir.
Okay. Now I would just actually now before it, I would like to request Dr. Chakrabarti, who says that he has some engagement. So Dr. Chakrabarti can continue his remarks in the beginning, and then I will start conducting the session. Dr. Chakrabarti, please.
Thank you. Good afternoon, everybody. Good afternoon, sir. For very ably taking over this responsibility of chairing the session. And I am a last minute entrant. I just before lunch only I got the invitation. Maybe a replacement for somebody. But then nevertheless, I have some responsibility. I felt because as a ADG plant protection, I have seen this crop, which has a big stake in ensuring and upholding the food security of the country. And rice is one of the most important staple crop of our country, besides several other Asian countries. And we not only export 20,000 crores worth of basmati rice every year, but equal amount of other rice also, non-basmati rice also is exported. But with my association as ADG plant protection and also with the Codex Committee on Pesticide Residue, I have seen this crop from very close point of view. And as very rightly pointed out, sir, in the beginning remark, you said that rice tumor virus, which used to be a very minor disease, now it is finding very important place in this northeastern country. Several biotic stresses they are emerging as stresses, and the policy issues affects its production and international trade in the country. So, nevertheless, we have several policies with respect to wheat and rice. We have to emerge victorious, and definitely we are poised to become highest producer in the world, maybe in due course of time. But there are certain concerns which are also emerging because of cultivation of rice or irrigated rice because of the shortage of water. Many stresses have emerged recently, which you highlighted due to maybe changing climate, due to maybe changes in the cropping pattern, crop intensification, and policy issues. Definitely, you have to the workers who are there in this area. They have to address certain policy issues which affects which will help government to take initiatives for making this crop very productive remunerative. Rice is next to cotton in using pesticides, and many stresses like which have come includes this false mud. False mud of rice has become really rampant, and I have been keeping a track for last 3 years in MP region and UP region. I am seeing that it is going on increasing every year. Somebody has to take and ring the bell, and we need to get up, because otherwise, if it enters the other eastern part of India and northern part of India, it is going to be a real havoc. Where I have seen now 60% of plants. I recently happened to visit a field in this Gorakhpur region, in this region, and 60% of the crop area was affected by this false mud, which is really alarming. And farmers do not know, policymakers, and then scientists' contribution are required. Where these mud balls, they are crushed along with the processed rice, and next year when they are sown without any seed treatment, they are going to increase into a very severe situation, so that we need to get an alarm and raise an alarm for development. Bakani disease, as you all know, here it has become another menace. Osmut, and I said seed rot and rice tumor virus has become very destructive. And Dr. Kalyan himself has done on work on some bloom blotch or bark cold area, gloomy, and some other bacterial diseases which are recently which have emerged, which are of importance. So dry seeded rice has increased in the area. This is one of the policy matters which has come. Dry seeded rice or direct seeded rice, which is there, which has a great problem, has increased biotic-stress, because weedy rice has become a real problem. It is becoming a real problem issue in eastern part and many other areas where this is going to be there. Weedy rice or red rice, it is the problem which is going to be there. So herbicide management, pesticide management has to be done. We have largest number of pesticide registered, but most of them, like for blast and some important biotic stresses like the bacterial blight and blast, which is tricyclone, it is banned by some country. Isoprothylene is another one which is again banned by USA. So for tackling these issues, we need to gear up. We need to review our policies. We need to have some more label claim on this pesticide and the newer ones, so that we should either non-pesticide management we have to highlight, because the problems are always there with the dry seeded rice. Certain root grubs and all those things have come. New nematodes have also emerged in rice. Clear field technology is one of the very potential area which we must try to make use of the potential of clear field technology, which is nothing but making or finding an SNP in a gene called acetolactate synthase or AHS gene with a single mutation, either by mutation or by mutagenization, or maybe gene editing, which is we are not recommending here, because we have a large germplasm. We will find many of the variants which are having this, which are insensitive to binding of the herbicide, and therefore we can have certain issues cleared with this. And then biomodifying our rice with many of the bacterial blight resistance which are already there, as well as Pica gene and PI genes for which are blast resistant. That is another issue. biotechnologically, again, there are many advancements which we need to see for biotic stress management. Arsenic is another problem which has come, and recently in CCPR now the PPV, which is 350 PPV has been fixed as a by Codex Committee or Contaminants Institute based on the data provided by India in Codex. FAO and WHO, they have fixed that value. However, we need to work upon to mitigate and to minimize the problems of arsenic. So that should not be another detriment or limitation to our growing concerns in trade. So these are the few things which immediately came to my mind, and I'm very grateful to you and to organizers for giving me some time out of your schedule. And also, as you have highlighted, I have some other ESRB related work going on, and some experts are already there. So I'm there. So I might have to go there, sir, and I will be joining in between. I'm sorry if I have not been there for all the time. Thank you so much, sir. Thank you, Dr. Mai, sir, and thank you, organizers, for giving me this opportunity. Thank you. Sir, your voice is muted.
अभी आ रहा है क्या वॉइस?
हाँ, अभी आ रहा है.
Now, friends, I would like to invite the keynote speaker, Dr. Finbar Organ. I hope he is here around. Dr. Finbar Organ is an ecologist, entomologist with a vast experience in the development of sustainable farming. His research focuses on reducing the negative impacts of agriculture while increasing farm profits. Finbar research mainly focuses on food production systems using agro-eco practices, and he developed holistic approaches for herbivore management on farm. I think he has a very vast experience in the research in areas of ecological management of pests, and I am sure that his lecture is going to be very useful on the rice ecology vis-a-vis the pest management. May I request Finbar Organ to please come on the mic. Finbar Organ.
Hello. Good morning.
Morning.
Okay. Thank you. So can I just check that that you can see the screen?
Yes, yes.
Everything is okay? Thank you very much.
Yes.
Well, firstly, I'd like to thank you for the very kind invitation to speak at your Congress, and I wish you the very best success for this first Indian Rice Congress. It's amazing to be able to get such a large group of people together during the COVID outbreak, and I hope you're all safe and keeping well, and that your work isn't affected too much. So in this talk, what I want to try and do is link the Millennium Ecosystem Assessment to IPM and to insects and pests and their services in rice ecosystems, and of course the services of beneficial insects, and to look at this as a way to try to better comprehend what's happening at an ecological level in rice ecosystems, so that we can then determine best ways to manage pests and to manage beneficials. And at the end, I'll try and introduce the concept of integrated crop management. Oops, sorry. To you. Okay, so why is it important to look at new ways to manage and ecological ways to manage rice? I think there's a bit of a problem here with my slides moving forward. One of the the main issues, of course, is that rice is such a large biome. It's larger than tropical rainforests, for example, as large as natural grasslands, with over 165 million hectares of rice production worldwide. And in South Asia, 62 million hectares of rice production. So huge areas of land, and it's really probably not feasible anymore to just keep dumping pesticides on the rice. If we look at the Millennium Ecosystem Assessment, and all of you will be familiar with this, we're focusing on biodiversity and how biodiversity contributes to ecosystem services, and these are services to human societies. And there are four categories of services that we're interested in: the supporting services, provisioning services, which of course includes rice production, but also includes other aspects of productivity in rice agroecosystems; the regulating services, which is what I'll talk about mainly today, and I think this is the key to understanding rice ecosystems; and the cultural ecosystem services. So the rice regulating ecosystem services, this is really the key to functioning in rice ecosystems. And it's the key not just to rice productivity, because regulating services will affect productivity. They'll affect weeds, for example. They'll affect water, carbon sequestration, climate regulation, etc. And what we're interested in is regulation in terms of how parasitoids and predators will manage pests. Regulation is a very simple concept. When population densities of a herbivore are high, sorry, my slides keep jumping forward. I
just sorry to interrupt, Finbar.
Yeah.
I think your slide has each slide has some certain time set.
Yeah.
So I have removed the time. Shall I share the screen?
Yes,
now?
please. Thank you.
Yes.
Thank you. And I'll just yes.
Kindly unshare.
Yes. Okay. Thank you. Thank you. So,
are you able to see now?
Yes, maybe slide
yes.
number 6.
Yes, 1 minute. So,
Right.
Thank
you very much. Thank you. Yes, I'm very sorry about that.
You can say next, so I will move the slides.
Yes, thank you very, very much. Sorry about that. I can now relax a bit. So as we all know, regulation, when populations are high, regulators will kick in and pull the population down to a lower level. And when the populations are low, the regulator is equilibrium density. And this is a density that doesn't really exist, and it can shift depending on the environment. But it's what we're aiming for. It's a central a tendency for the populations to stay level. And this is our negative feedback mechanism. And what I want to show is that these negative feedback mechanisms exist in natural systems. And when we move from a natural system to a human-dominated or a system where we're trying to control the functioning of a system, we're still governed by all these negative feedback mechanisms. So you can move to the next slide, please. Thank you. So if we think about a rice monoculture, or a large field of rice, it doesn't have to be a huge monoculture. This monoculture is governed by all the same natural regulating systems that exist in a natural biome. So if you take, for example, the case of an introduced weed, water hyacinth, for example, when water hyacinth is introduced to a new region, it very quickly takes over and it reaches the carrying capacity of the ecosystem. And normally that's the space that's available for the plant to grow on a lake surface or on a pond. And to control that, you might introduce a biological control agent. And for example, with water hyacinth, the water weevil, the mottled water weevil, has been a very successful biological control agent, reducing the densities of the water hyacinth. And it's the very same thing with rice. We've planted rice, we've dominated our system with rice. We want nothing else in the system except rice. And so what happens is the ecosystem's natural regulatory mechanisms start to come into force. Next slide. And what are these regulators? Well, actually, in this case, the regulators are the herbivores. So the natural system will turn on the regulation by herbivores to try and knock down the biomass of rice and the population densities of rice. And this is just a natural phenomenon that we always have to fight against. So rice stem borer, for example, works the same way as a water weevil. Is just going to attack the stems, try to kill the tillers, reduce the output. And of course, this is good for natural systems. This is what a natural system aims to do, to increase biodiversity and to increase productivity in a natural context. But it's going against what a farmer wishes to do. Next slide, please. Yes. So what we have then is in the natural system, because there's a whole series of negative feedback mechanisms, we then have regulators that are working to keep the populations of these herbivores down. So we have two systems of regulation going on. One is where the herbivore is trying to regulate the plant, regulate our rice populations, and the other is where the natural enemy is trying to regulate the herbivore. And what we want to do is we want to optimize one and reduce the other. And the key feature of population regulation to get that negative feedback mechanism working is that we must find density dependence. Now, this is just data from 15 rice fields in the Philippines. And you can see here the density of two herbivores. That's the whiteback planthopper and the brown planthopper, and the corresponding densities in those fields of a range of different spiders. And you can see there, in both cases, a tendency, quite a strong tendency for density dependence. This tells us that spiders in the rice field, in these 15 rice fields at least, are regulating our herbivore populations. Next slide. In the same fields, looking at myrid bugs like Citerinus, we see the same thing. As the density of the herbivores increase, the density of the predators also increases, indicating that, again, that this species is likely involved in the regulation of our herbivore species, these two species in particular. Next slide, please. And again, in this case, although the data here is not that strong because it's coming from sweet net samples, we can see the same thing with Anagrus, which is an egg parasitoid. As the population densities of the two herbivores increase, the densities of free-living adult parasitoids also increase. And if we were to look at eggs using sentinel plants, we'd also see density dependence in this case. So these are all the herbivores that are in the field that we know are contributing to knocking down the densities or the populations of herbivores that are feeding on the rice. Next slide, please. Now, what I'm going to try and do now is overlay IPM and ask some questions about where IPM is going. So we know that IPM has a number of objectives. In fact, IPM was developed in the 60s as a response mainly to an overuse of chemical pesticides and to the problems that overuse was causing in the environment. It was really a response to Rachel Carson's book, Silent Spring. The objectives of IPM are to reduce, of course, the economic losses to crops from insects and to reduce pesticide use. And this is very important. The objective, one of the main objectives of IPM is to reduce pesticide use. And how do we do that? Firstly, we exhaust all alternative environmentally friendly control options before we resort to pesticides. And it's important to note that the pesticides are used only as a curative measure. So first we look at cultural controls, physical controls, biological controls, yeah, and as a last resource, we look at pesticides as a curative measure. There are two main ideas that govern our IPM. One is that different control options should be integrated to function as one management system. And the second is that we must monitor the insect densities we must base our control actions on the threshold densities of pests in our fields. And I think everybody knows this very well. Next slide, please. But now I'd have to ask, is IPM failing? And I think there's a lot of evidence to say that in general, IPM does not work anymore as we had planned. And I've seen this in many of the different countries where I've worked and on many, many different crops. And I'll give you three main reasons why I think this is happening. Firstly, if you look at the objectives, and one of those objectives of IPM was that pesticides should only be used as a last resort. What we're seeing now is in many, many cases, it's not used as a last resort. It's used as one of the first control methods that farmers will turn to. And now it's increasingly being used as a preventative measure or in prophylactic approaches and not as a curative measure. So that's the first failing of IPM. Here we see where normal practices do not meet the requirements of IPM. Secondly, monitoring is an issue. Farmers in general do not monitor insect populations or do not know how to interpret the populations that they see in their fields. And thirdly, we have huge problems with the establishment of threshold densities. So you only have to look at the literature on leaf folders to see that there are a wide range of threshold densities being spread around, and it's very difficult to know. Sorry, it's very difficult to know which ones to follow. In fact, if you look at the threshold densities for some insect pests, you'll see that it's impossible not to use chemicals based on the densities that some researchers are proposing. Okay. And then you can add that in many cases, IPM doesn't work because many of the different control options do not work synergistically, but are actually antagonistic. Next slide, please. So what do? We have to, I think we have to rethink our approach, and we have to look at the rice ecosystem in terms of optimizing the regulating ecosystem services, the top-down regulating ecosystem services that are coming from our biological control agents, like our predator.
Please unmute.
Hello?
Yes, you
Yes.
are audible.
I'm audible? Okay, sorry.
Yes.
You get muted actually in meeting.
Oh, okay. So how much did you miss? I'm not sure. Okay. Okay, so I'll just continue on. I'm not sure where. Okay, we'll just go to the next slide, please.
I think it's okay. I think the same slide. So everything is okay.
Oh, okay. So what I was saying there, I'm not sure when I was muted. What I was saying there is what we really need to do is rethink how we approach the rice ecosystem and its management. We want to reduce the regulating disservices, because there are regulating disservices, and we want to optimize the regulating ecosystem services. These are the top-down regulating services imparted by natural enemies in the rice ecosystem. Okay, and we'll see as I move forward that very often we're doing the exact opposite in our rice ecosystems, and this is causing tremendous problems. Next slide, please. Okay, so for example, if we think about regulating ecosystem disservices, one of the things that we're doing is we're increasing the carrying capacity, or we're maximizing our rice productivity to sort of saturate the carrying capacity of the environment. And this, of course, is going to have knock-on effects, because the herbivores are just going to chase after that productivity and try and knock it down naturally. Okay, and here you can see this is data, and this is quite amazing data from Indonesia, and you can see here that at the bottom there in Central Java, many farmers are trying to achieve 7 rice crops in 2 years. So in this particular year, when we did mapping, they're getting 4 crops in a year. So in tremendous productivity, and of course, this is going to increase pest populations dramatically and disease incidence as well. How do farmers do that? You can see there's an overlap in the cropping. So if we go to the next slide, you see that what's happening is many of the farmers are setting up their seed beds at the same time that there's a standing crop in the field. So it allows them to overlap 2 crops at one time, because of course, when they transplant the seeds, they're filling out that area from the rice that they've just harvested. And as you can see in this seed bed, these seedlings are quite old already, quite advanced. And this system is allowing the farmers to get 7 crops in a season. And of course, it's a disaster in terms of pest management, requiring a huge amount of chemical pesticides to control things in the field. Next slide, please. The other thing that's happening, and this sometimes is unplanned, is you get a ratoon crop that carries over the pest incidence from one cropping season to the next. So if we go to the next slide. So this is data from the Philippines. This is near Laguna Lake in Luzon. And these shapes at the top here, are polygons, are maps of a rice growing area. And every month of the year in 2013, the stages of the rice crop were followed. And what you can see there is that although the farmers think that they have 2 separate crops in the season, because of a lack of synchrony of cropping, and because some of the areas have saturated soils and you get a ratoon crop, well, what really is happening there is that the farmers have a continuous cycle of pests increasing throughout the year. And the only time that they get a break in the crop is actually in November, when the ground is too dry for any rice to grow and for any ratooning to occur. So a lack of synchrony and ratooning transforms 2 rice crops into one continuous presence of rice in the field. Next slide, please. So just here with these red arrows at the bottom there, you can see that if you look at different parts of this map where there's a farmer has a fallow period, for example, to try and cut down pest incidence, as soon as he transplants, those pests will move from ratoon areas into the fallow, or from a standing crop into the recently transplanted crop. Next slide, please. The other thing we do, and this is really a method to increase the carrying capacity of the environment by us, the farmers, manipulating the environment, is we add fertilizer. And of course, it's very well known that as you increase fertilizer inputs, you get an increase in the incidence of many pests. Now, it's interesting that when we've looked at nitrogen content on soil in rice in the Philippines, and we've looked at the incidence of different pests in a pesticide-free scenario, what we find is actually the opposite, because the regulators, the natural regulators, the predators, will actually knock down those pests and do so as efficiently at high nitrogen levels as they do at low nitrogen levels. But basically, if you think about what's happening here, we're increasing the carrying capacity of the environment and thereby increasing the infestation of the pests. Next slide, please. Here's a very well-known case with whiteback planthopper. So in China, it's known that when hybrid rice was adopted, particularly hybrid rice with the cytoplasmic male sterile parent, and planted and adopted by farmers throughout China, there was a corresponding rapid increase in the incidence of whiteback planthopper and the incidence of planthopper outbreaks. Okay, so in this case, we again increased the carrying capacity of the environment for rice, and we reduced the natural mechanisms in the rice field to counteract the growth of whiteback planthopper populations. Next slide, please. Okay, so what can we do to now reduce the regulating efficiency of the herbivores on the rice. And what we're trying to do here is we're trying to knock the herbivore out to some extent. And one of the things we do there is, of course, host plant resistance. This is the most popular approach, the one that most scientists have concentrated in rice over the decades, and it continues to be at the top of all the research that's going on in management of rice pests. And this is, of course, you'll know, the brown planthopper. So we move to the next slide, please. So we know the story. Here you have rice plants, Ratu Hanati, which has the BPH32 and other genes with resistance to the brown planthopper on the left, and IR62. Okay, and IR62, which is a derived rice variety on the right, again with the BPH32 gene. And the pots there, the red pots, have been infested with 25 planthopper nymphs, and these nymphs were allowed to develop. And you can see that the plants successfully defended themselves against the nymphs. So host plant resistance, what's it doing? It's upsetting regulation by the herbivore of the rice population. Next slide, please. Okay, and this is some data from the field, and you can see there with the red and the green marks on the left in graphs A and B, 2 years of data from the Philippines. You can see IR22, which is susceptible, IR62, which is resistant. You see how the IR62 variety has consistently low densities of brown planthopper compared to the IR22. And this is occurring irrespective of the contents of nitrogen that's thrown on the field. Of course, IR22, the hopper is responding to nitrogen and increasing its population densities. Next slide, please. But we all know the problem with host plant resistance, and it is a very serious problem, and that is that the planthoppers very rapidly will develop the capacity to feed on resistant varieties. So we all know the story, I think, of Mudgo. So here you have Mudgo. Again, the red pots are plants that have been infested with brown planthopper. The green pots are control. And you can see that in spite of having a very strong and perhaps one of the strongest resistance genes, BPH1, these plants have been overcome by planthoppers. So there's limitations to this technology, and we have to face those limitations. We have to recognize those limitations, and we have to work against those limitations. And one of the ways to do that, of course, is to increase the different strategies of control that we use in the rice ecosystem. Next slide, please. The other, one of the approaches has been to say, okay, to try and stop the breakdown of resistance, we will pyramid genes. And this, again, is the most popular approach to prolonging the durability of host plant resistance in rice. And one of the problems that we see here is, firstly, we know very little about how genes work together in rice against pests. But the other is that we can see that there is, and this is only logical, there are trade-offs between the strength of resistance and certain aspects of rice plant growth. In this case, these are resistance genes against the green leafhopper. We can see that there's a trade-off between resistance and the number of loci that we've pyramided into the plant and grain weight. Next slide, please. So this is just reiterating that as we increase resistance by pyramiding genes, it's only logical that we'll get a reduction in yield. And the other thing we have there is nitrogen. We can try and optimize nitrogen applications to try and optimize yields, but reduce herbivore pressures. Can I go to the next slide, please? And I'll just go to the next slide as well. Yeah. So again, going back to that same study from the Philippines, we still know surprisingly little about how nitrogen affects the whole community of insects in a rice ecosystem. And this slide here indicates that nitrogen has a very important role in increasing the tolerance of rice against certain pests. And if you look there at graph I, IR62, it's a resistant plant. when you throw nitrogen on it at high nitrogen levels, you're actually getting overcompensation. So the insect in this resistant plant is actually inducing a higher productivity of grain. So there can be some positive outcomes from fertilizers. Next slide, please. And again, in this slide, again from the Philippines, from the same study, we can see that many of our specialist natural enemies are responding to the higher nitrogen levels and are even more efficient at high nitrogen levels, knocking down our pest densities. So these are all beneficial things. So it's showing us that the negative feedback systems are able, if they're intact, if we allow them to operate, they're able to counteract perturbations. And the perturbation that I'm looking at in this case is an increase in nitrogen applications. Next slide, please. So the second part of that, I've so far been talking about how we can counter the regulating ecosystem disservices of herbivores. Now I want to look at how do we optimize regulating ecosystem services of top-down predators and parasitoids. Next slide, please. Here we're, am I okay? Here we're doing not so well. And part of the problem is this massive increase in pesticide use that we've been seeing all over Asia since the 2000s. And we know that this is affecting herbivores. Next slide, please. This again, data from the Philippines, and you can see that in insecticide sprayed fields, we get a knockdown in important parasitoids like these egg parasitoids, Oligosita and Agrus, in our fields. Next slide, please. So as our natural enemies are being knocked down by insecticides, what's happening to the herbivores? Well, in many cases, they're developing resistance, and there are many cases, well, where the insecticides induce them to become fitter and to reproduce even more. And this is very well documented, for example, with plant hoppers, but also occurs with stem borers and leaf folders. Next slide, please. This is just an example again of resurgence. You see here how the pesticide has knocked out natural regulators just on this specific field where the pesticide was applied, and the herbivore then can reach the carrying capacity of that field, which means it completely destroys all the rice for that farmer. Next slide, please. Now we can also try and improve biocontrol by augmentation using biocontrol agents, but this is expensive and it's often difficult to achieve success with biological control in large field crops like rice. Next slide. So I guess where people who are interested in ecology and ecosystems and regulation, et cetera, are going is we're looking now towards farmers and land managers giving services back to the ecosystem. And so we talk now about services to ecosystem, and there are, just like the ecosystem services from the Millennium Ecosystem Assessment, there are 4 services to ecosystem that we're interested in: protecting services, restoring services, and their underlying supporting services, which are mainly, policy or cultural approaches, etc. Next slide, please. So we've been working with ecological engineering in the Philippines. Am I okay for time? I'm okay. We've been looking at ecological engineering in the Philippines. I'll just rush through this very quickly. Next slide, please. And what we're trying to do there is we're not only trying to enhance the regulation efficiency of specialists and generalists, but we're also trying to increase resilience. Now, if you think of resilience in a system with many components, where you have more connections between the different components, we're getting a more resilient ecosystem. And this comes from network theory. Okay, next slide, please. Okay, so this is so this is just data from 6 sites that were spread right throughout the Philippines, and we can go to the next slide. And this unusual looking field is an ecologically engineered field with rice patches where we've tried to induce or increase the biodiversity of organisms in general in the rice fields and to see what happens in those systems. Next slide, please. So I'll just very quickly go through this data here. So there's a number of graphs there, 2 panels of graphs. At the top we have parasitoids, and at the bottom we have predators, 2 predators. And on the left we have fields, conventional fields, and on the right we have fields with vegetable patches. Next slide, please. So what you'll see straight away is that in fields where there are vegetable patches, we have a much higher diversity of parasitoids. And this is important. We're not just talking about abundance here, we're talking about a diversity. So we've increased the complexity of the ecological network, and this brings about resilience in theory. Next slide, please. And you can see also we've increased the abundance of the 2 predators by having these vegetables growing in the field. Next slide, please. Now I just want to sort of show you this recent data. We just published this paper recently, looking at how the brown planthopper and the whiteback planthopper, if you put them together on plants and you increase the temperature, so trying to look at what happens under climate change scenarios, the 2 herbivores together actually will combined have lower populations than if BPH occurs on its own. So here's a situation where competition between 2 species is reducing the overall populations of the 2 species under elevated temperatures. And this is just one example of how increasing diversity can bring about stability and resilience in the rice ecosystem. So even though we're talking about herbivores here, having 2 herbivores is often not a bad thing. Next slide, please. So the bottom line of all those studies on ecological engineering was we had increased productivity on the farms. We had, of course, reduced pesticide use. We had better regulation of our herbivore pests. We had increased biodiversity of things like birds and parasitoids, pollinators, and we had even reduced rat damage in the fields. Now, for these things to move ahead, we need a change of frame of mind. We need to work on our supporting services to ecosystem. And these supporting services to ecosystem come from culture, come from religion, come from politics. And they're the things that guide people to their decisions on the rice farm. Next slide, please. And what I think we need to do now is we need to move away from integrated pest management, and we need to move to a new paradigm. And I think that the paradigm that fits where we're going at the moment is integrated crop management. Now, this paradigm has been around a long time, but why I like it is because it is holistic. We can't anymore just focus on controlling one pest or one disease, but have to look at things together. And it's also depends on the rice manager or the farmer being conscientious about the environment and looking towards a holistic approach to minimizing pollution, minimizing the use of chemicals, and enhancing the efficiency, ecological efficiency of his system. Next slide, please. Now, one of the ways we can do that is work through these sustainable crop platforms. This one is very well known, SAI, based in Switzerland, and I know there's platforms like this in India, and platforms specifically for rice, so the sustainable rice platform, for example. And these are governed by consumers and multi-stakeholder consultations. And this is a step forward, I think, in developing these supporting systems, supporting services. Yeah. If you think about IPM was based on economics, and it was based on thresholds. But these integrated crop management approaches are based on protecting the environment and on cultural sensitivities. So I think it's a move forward, and I think this is where we need to be going next. Of course, there's still problems that need to be ironed out in these systems, but this is where we need to be going. Next slide, please. So just in conclusion, I think we need to start to think about a change in our paradigms and where we're and how we approach pest management, and we need to reflect more the functioning, the ecological functioning of our rice ecosystems to better understand the approaches we must take to reducing pest incidents and increasing productivity. Thank you very much. Next slide. Thank you very much. Okay. I can't hear.
Dr. Sidimai sir, your voice is not coming.
Are you able to hear now?
And now it is okay.
Yeah. Thanks very much for the nice lecture. Now I will call upon Subbaralu Mohan Kumar, who is a university rank holder of ICAR, giving the lead lecture. He is actually an entomologist having a postdoctoral training in molecular genetics under Rockefeller Foundation program in Rice Biotechnology. He started his career at TNAU and he has been coordinating multi-disciplinary projects there. I request Mohan Kumar to kindly take the floor.
Yes. So, respected chair, am I audible?
Yes, yes.
Yes. Okay. Respected chair and distinguished co-chairs, Dr. Sidimai sir and Dr. Chakravarti sir, and the conveners as well as the rapporteurs of the session and the good speakers of the session, both friends of us, Dr. Finberg as well as Dr. Bentur. I am very happy to be with us in this session for the Biotic-stress, and good evening to you all, and good morning to Dr. Finberg, and thanks for the wonderful opportunity given in the first the Indian Rice Congress, because so we attended the National Rice Biotechnology Conference for 7 or 8 times. This is first time, especially for a good stable crop, we are conducting the first Rice Congress, and thanks for the nice efforts for giving me the opportunity to share my views on the post-harvest pest management after Dr. Finbar on the pre-harvest pest management. So can I get my slides, please?
Yes, sir. I'm sharing.
Can I share from my window?
Yeah, you can also share, sir. No problem.
Some
Please.
background sound is coming. Just
Sir, if you have speakers, headphones, kindly your voice is disturbing.
yes, yes.
Are you able to hear now? Hello?
Yeah, now, but again, the sound is coming.
Okay. Just wait a minute. So, is it clear now?
It's clear now.
It's better.
Okay. So, please.
Sir, you are sharing or shall I share, sir?
You can share, sir. You got
Yes.
it?
Yes, yes.
Okay. Sorry for everyone.
So you are seeing the slides?
No, sir. It shows that you have started screen sharing. Can I go ahead?
Now, sir?
Yes. Now, yes. Yes.
Yes.
So next slide, please. It's already time. Next slide, please. So India is a good producer for the rice, as well as nowadays the storage at the farm level is reducing after the combine harvester, and the farmers are selling the produce immediately to the either the processor or to the regulatory agencies or the especially the Food Corporation of India, etc. So in that, the storage is mainly on the either on the processing side or in the godowns of the Food Corporation of India or Central Warehouse Corporation, like that. There are so many storage houses, and the food grain loss is around 12 to 16%, whereas it is accounts to be by the insects around 17 billion Indian rupees. Next slide, please. Next. So in that, just if you see that, oh sorry, for the previous one. So in that, the storage capacity is around 28 million tons. So in that, if you see in those godowns, there are so much of food grains are stored to have a buffer stock, so that it may be in the godown for 6 months or 1 year. Sometimes it is allowed to keep it for more than a year, depending upon the situation. And also they are transported to various public distribution system. So the storage is very much in the Food Corporation of India, and in the both open storage as well as the especially they call it as cover and plinth, the cap storage, and also in the godowns. And now there is a transition to silos also. So slowly they are establishing the silo. One of the example is in Moga in Punjab as well as in Kaital, Haryana, and we also have it in Coimbatore. Next, please. So like that, the food grain storage system is very well developing in the various levels to minimize the losses. Next slide, please. So in that, there are 15 to 20 insect pests are associated with that. Mostly as Dr. Finbar told, the preventive methods are taken very well in the storage godowns, especially only with the chemical insecticides, especially the malathion, deltamethrin. So earlier with the dichlorvas also, and the curative method after seeing the insect infestations as 2 number per sampling. So they are able to go for the fumigation with the phosphine. And these are the important methods used, that even though we are calling that as integrated pest management, etc. But in storage, mostly either as a preventive methods with insecticides or with the curative methods with the fumigation of the phosphine is done. Can I get next slide, please? So while doing that with the various type of prophylactic methods, and also they are making the various fumigants, especially earlier they used the methyl bromide, but now so it was banned. So they are able to have only one alternative, that is the phosphine gas, because it is effective against all stages of the insect pest in the grain storage, and also it is cheaper, easy to apply, and one of the important advantage is residue free, and it doesn't affect the viability of the seeds also. So various godowns, they are using the tablets of the phosphine for the control of the insects. Next, please. Next, please. So in that, there are various type of tablets are available. So now even in the sachet packet for the low scale storage, the phosphine tablets are available for small scale storage. Next, please. Next, please. So in this, if you see the mode of action in the various type of oxidation reduction reaction which is present in the cycle, and reducing the oxygen content is most important for the control of the insects. Especially if you heard about the hermetic storage, the International Rice Research Institutes also developed the super rice bag. So in that, mainly the oxygen level is reduced, and so that the pests are not able to thrive. And because this is for only a small scale storage, whereas for the large scale storage, either a bag storage or the cap storage, so they are only applying the phosphine. So in that case, they are interrupting in the respiratory pathway in 4 important places, and in that, the one of the important enzyme which are regulating that is the dihydrolipoamide dehydrogenase. So in simple, we call it as DLD gene. And this gene is more important. So it is a flavin-dependent oxidoreductase, and that is with the help of the cofactor FAD, they are able to transfer in the electron transport chain of the phosphorylation. And in that cycle, if there is a disruption, the respiratory arrest is there, the insects are able to killed by the phosphine. So in that, if there are any mutation in this DLD gene, so there is a resistance development in case for the phosphine. Next slide, please. So this mechanism was very well characterized in Australia, University of Queensland. We are able to get the collaborative projects with them, and we are able to work on these fumigations in the especially bulk storage, bulk grain storage areas. And improper storage is an important one for many of the godowns. They have the cover sheets having the holes, or just to overlap sheets are used. There is no the controlled atmosphere, and also improper sealing and operating procedures are used. And the movement of the stored grain also occurs because we are transporting the various places from Chhattisgarh to Tamil Nadu, from Andhra Pradesh to Tamil Nadu. So there are a lot of rice grain transport is there through the rail wagons, and in many of the cases, the stored grain pest also transferred from one state to another. Next, please. And as like that, the international also sometimes we are exporting the non-basmati as well as the basmati rice, and there are a lot of possibilities. And also we are importing the wheat grains, and then so there are chances. And in many of the cases for the toxicity, so it is depend upon the concentration as well as the exposure time is more important, and also the sensitivity of the against the particular insect, and also how much grain temperature it is present, because it only allow them to for the multiplication of the insect. So in these cases, now we are moving slowly from the back storage to silo, and there is no perfect method done for the storing the rice grains in silos, because there are a lot of caking, the temperature is moving ahead, so that even 1 degree increase in the temperature, the multiplication rate is very high. So we have to develop a very good method for the storing the rice in the silos. But in the back storage, we are applying the phosphine. So what happens to that? Next slide, please. So in that, it is a very big challenge. We did the systematic survey throughout India, except a few states like Jammu and Kashmir and Northeast. And so all other places, we did the survey in most of the storage houses of the FCI or CWC or State Warehouse Corporation. Next, please. And we found that the most of the godowns, next, please. So the various type of resistance occurs, and in that we put the two doses as a discriminating dose. One is low dose and the high dose as like prescribed by the Food and Agriculture Organization FAO protocol. And based on that, if it survives in the low dose as well as in the high dose, it considered as a susceptible, whereas on the reverse, if it survives in the low dose and the high dose, it is considered as a strong resistance. So earlier strong resistance was reported by Rajendran and his co-workers in India in different pests around 300 to 380 fold. And our systematic survey, next please. So our systematic survey showed that, so in various states, so it ranges from 30 to 100% resistance, even at the higher concentration in the tribolium. If you see that there is a 31% or 30.33% to 90% is the resistance occurs in case of the Tribolium castaneum for various type of stages, immature stages, which are present in that in case of the phosphine. So the continued exposure to phosphine lead this scenario in our country. Next, please. And not only on the Tribolium, then next also for the Cytophilus voraceae. So there is a resistance, but less level as compared to Tribolium. Next, please. Next, please. So just if you see that, is there any possibility to analyze at the molecular level? As I told, the DLD gene is responsible for that, and we are able to study the molecular genetics of the phosphine resistance and see the mutations which are present in that. Next, please. So we are able to say that the resistance alleles are present in the various kind of populations collected from different states. So if you see around 660 insects tested in Andhra Pradesh, so the R allele is present in almost 97.43% in case of the Tribolium castaneum. So 74% as R allele. From the heterozygotes, if you see that at least one R allele, if it is present, it is 97.43. Like that, almost more than 80% of the R alleles from different states, so they have the resistance for the phosphine. So next to that, not only in Tribolium and also in the second pest called, next slide please. So the Cytophilus, this is the Tribolium population. So you can easily see with the help of a CAPS marker. So we are amplifying with the DLD gene, and then we are doing the restriction digestion, and PCR RFLP shows that, so the able to differentiate the heterozygotes as well as the homozygotes, either it is susceptible or to the resistant, and we are able to see in both the Tribolium, Rhizobertha as well as in the Cytophilus. Different kind of enzyme we are using it for different pests, and in that it is able to give the very good. Just if you see that this is a heterozygote, whereas in the resistant, whereas in the susceptible it is uncut. So like that, it is possible to differentiate based on the PCR RFLP. This is also one of the method can be used for phosphine resistance in guidons. Next, please. So same thing. So for the insecticides, there are many synthetic pyrethroids are used, especially the deltamethrin is a common use in many of the guidons, not on mixing with the grain. It is on the alleways as well as on the back surface, and they are spraying on the windows as well as the doors. And in that also, because of the continued spray, they are able to have the resistant allele. And in that, we are able to find a mutation in 929th position for this, the deltamethrin. And this is, everybody heard about the knockdown resistance KDR, and the super KDR as recorded in other diphtherians as well as that is like mosquitoes. We are able to find this Cytophilus verae, this mutation, and that is related to resistance. So this is one of the good work of my PhD student and Sonu Kumar Singh. Next, please. So based on that, we are able to see the various kind of resistance to phosphine also. So we have to
Dr. Bhuvankar, you have 4 minutes more.
Yes, sir. Yes, sir. I will go. Okay. So thank you, sir. And just if you see that the only treatment now they are applying either as a prophylactic with the help of the deltamethrin or malathion, and already the malathion resistance was well known. And in that case, the field populations, how to go for that, and we have done with the various kind of insecticides. And in that, one of that, even though there is no label claim, only in the laboratory study, we are able to find that the chlorpyrifos methyl 0.5% was effective against the storage pest. Next, please. Not for mixing with the grain, just only for the surface treatment. So we are able to see just spray it on the gunny bag and allowed the insect of the Tribolium, Rhizobertha, as well as Cytophilus, as well as Trochoderma granarium, and we are able to get more than 96%. And this kind of insecticides, even though if you are adding with the synergist, the resistance level are not broken down. And especially the PBO or Pungam oil or various kind of the synergists we have added and see that, but the efficacy was not as much as like that of the chlorpyrifos methyl. Next, please. So the next to that, when you are going for the novel method, so monitoring is also important. Besides that, is there any other method as like the hermetic storage? There are other methods that can be combined with this fumigation so that we can at least delay the level of resistance development. So it will be an effective one. So we are able to make, in case of the phosphine, so extension of the exposure time. Next slide, please. extension of the exposure time from 5 days to 7 days, we are able to get the good control. As like that, the concentration also increased in case of that, so it also gives. But these are all the temporary measures for the next, because based on that, the CWC increased the time from 5 days to 7 days now, exposure time. But depending upon the population, the efficacy varies. So we have to manage with the available one. This is a very good fumigant, but if you are using proper fumigation, we are able to reduce the resistance development. Besides that, there are different type of silos. Now they are moving towards that, and that like a concrete silo, steel silo, even Indian Institute of Technology in India also developing a farmer friendly silo with a local small and marginal farmers. And in that, how to store this is very good for the weed system. But for the rice, as a rice grain, it is very hard to see that there are a lot of the fine tuning is necessary for different type of silos. And this is also, and these are all the silos which are in various Western countries, especially the Australia and other places, they are using this. And India also, in few places, the private functionaries, they are making either a concrete silo, and now they are moving to steel silo also. Next, please. And this is where our technology intervention should be there to move from cap storage, that is the open storage, to silo storage. And also for trapping or monitoring the insects, we can use different kind of traps. So in that, I think I am Mohan Kumar, and there is another scientist retired, Dr. Mohan. Everybody knew about the entomologist from TNAU as Dr. Mohan. He called as Trap Mohan. Okay, so he developed different kind of traps for monitoring the various kind of storage pests, probe traps, ultra UV light traps, as well as various type of egg removal devices. We will see it in the next few slides. And he is one of the partners in various type of projects. We did work with him. Even now he is continuing. And those who are, next slide please. And those who want to see the various aspects, you can go to www.mohantrap.com. So he is very passionate, and we also tried with various type of pheromone traps and the aggregation pheromone and Indian meal moth and rice moth. There is a sex pheromone also available. So we tried, and it works very well for the red flower beetle as well as the Rhizoporta dominica. But for the rice weevil, it doesn't have the much attraction for the aggregation pheromone. Next, please. And so the probe traps are used in bulk storage as a monitoring one, just to see the various kind of movement. Next, please. Similarly, you can use this.
Now. 1 minute.
Yes, sir. I'll finish, sir. So the probe traps are used. Different kind of traps are listed. Next. Next. UV light trap. This is used by the Food Corporation India. This is an important device, egg removal device, and that the adult also reduced. And this is useful for various kind of removal of the insects from the rice, and it is used, and also we are exporting to other countries. And this machine, based on the principles of impact, smooth brushing and sieving as well as the blowing. So this is an important device. Next, please. So the seasonal flight activity we have seen with the pheromone in the production area and in the semi-cropping area as well as in the storage area, and very low numbers in the Rhizoporta dominica and the S. veraceae is present in the 3 habitats, whereas Tribolium is the big menace, and the trap, the movement also is present, and also the catches in which we got it from different places. We also studied the gene flow of the DLD gene, how it varies. Next slide, please. So I'll finish it in 1 minute, sir. Okay, so these are the seasonal flight activity in different part of the year from June to May. And just you can see that in the storage down area only the population is very high when compared to the production area and the semi-cropping area. And the flow is the movement is also very much restricted only with the storage down area, which is around 6 kilometers apart from the production area. Next, please. So the novel fumigation, as I told, so the covering and also monitoring the pathogen concentration is more important, and trapping and monitoring with the different life stages of the insects are important, and placing the tablets, and also the professional, skilled, trained persons are essential to do the fumigation, to do the pressure monitoring. So all kind of things, so that only we are able to achieve the good control. Next, please. Next, please. So the field fumigation, we are able to reduce the dosages. Next, please. So combination of CO2 with the phosphine also gives the good one, and the spinosad and sulfuryl fluoride also considered as an alternative for phosphine in different countries, but it has to be verified and in Indian conditions. Next, please. So these are the research publications based on our work in the Journal of Stored Product Heredity as well as in the Journal of Pest Science. Next, please. Next, please. So these are some of the extension activities we have done, and I also thank all the collaborators, Dr. Paul Ebert, as well as Dr. Manoj Nayak and Dr. Mohan and my student Sonu, as well as so give just time for us, and Dr. Chitra, Dr. Subramanian from IRI, and also for the various private firms, so that is from the UPL. So they also joined with us in educating the students as well as in educating the farmers in various kind of modern scientific storage of the food grains. Thank you for giving me this opportunity to share our views. Thank you so much.
Thank you, Dr. Mohan Kumar, for an excellent exposition of a grain saved is grain produced. Thank you very much for a very nice presentation. Now I would call upon the next lead speaker in this particular session, Gopalji Jha. I am sure that he must be around here. Gopalji Jha.
Yes, sir. I'm around, sir.
Yeah. So Gopalji Jha is a staff scientist 5 at the National Institute of Plant Genome Research in New Delhi. He was also a visiting scientist in Sainsbury Laboratory of John Innes in UK. He has worked in CSIR Institute of at Palampur and received several good awards. INSA Young Scientist Award. Then he has also been a gold medalist at Guru Nanak Dev. I think there are lots of things which can then, mainly his focus has been to study some basic research on mechanisms of really host pathogen interactions, maybe an infection process as well as process of resistance. So I think he'll talk on dissecting plant microbe interaction for holistic management of rice diseases. So I welcome you, Dr. Gopalji, and kindly take your time. But we have time up to only 4:30. So I think, can you take about something like 15 minutes so that we have time for discussion?
Yes, sir. Can you see my slides, sir?
Yes, yes.
Yes. I will try my best to finish in time. So thank you, sir, for nice introduction. And I'm thankful to the organizer, especially Dr. Kalyan Mandal, to give this opportunity to talk about my work. The title you have already spelled out. It is Dissecting Plant Microbes Interaction for Holistic Management of Rice Diseases. The previous speakers has talked about integrated pest management and post-harvest disease. So what I will do is I will try to take you through how understanding plant microbes interaction is helpful for management of the rice disease. You all know rice is susceptible to various diseases. I have just mentioned few here, and few has been already discussed during the previous presentation. So you all agree that the pathogen is very diverse. There are nematodes, there are fungus, there are bacteria, there are viruses, and their mode of pathogenesis is also very diverse. Some are biotrophs, some are necrotrophs, and some are in between. So handling this disease is a challenge for sustainable rice cultivation and global food security. And so understanding the mechanism by which the pathogen cause disease and plant defense themselves is an interesting area of research. And these research are having important practical implications for ensuring food security. So I had an opportunity to work on Xanthomonas rice interaction during my PhD. So I will just take this as a model to highlight how the host pathogen interaction is helpful to understand any pathosystem. I am referring XOO as Xanthomonas oryzae pathovirus. We saw that the Xanthomonas uses type 2 secretion systems, and there are other virulence factors which is required to cause disease in rice. However, rice recognize the action of these virulence factors and mount a strong defense response. However, being a successful pathogen, the pathogens has a strategy to suppress this induction of defense response, and mostly they do using the type 3 secretion system, but the plant has resistant genes which mount the action of these factors to override the suppression of the defense response. So this work I did it during my PhD, and subsequently Dr. Swanti's lab has been pursuing it, and I have just cited some of the references. But I will just like to emphasize that this schematic is almost followed in any host pathogen interaction, and our interest is to understand how a pathogen causes disease in plants and how plant responds against themselves. So now I will take you through another model which I have been pursuing at NIPGR. This is sheath blight disease. I told you that there are resistant genes which is used to modulate the pathogen to prevent them to cause disease. However, for sheath blight disease, there is no resistant gene known. So it is caused by a necrotrophic fungal pathogen, Rhizoctonia solanae. I have just put characteristic pictures just to emphasize how the disease looks like. And it is really a very severe rice disease, and the loss can go up to 70%. And I had said there is no resistant rice lands available for sheath blight disease. So disease is managed mostly through fungicides. So controlling sheath blight disease in an eco-friendly and sustainable manner remains a challenge. So our lab has been working on this, and we have various aspects of our studies. One of the main our study point is to identify important pathogenicity determinants of Rhizoctonia solanae and developing a strategy to silence them during infection process. Also, we are coming up that there are certain susceptibility factors in the host that is modulated by the pathogen to cause disease. And if we can somehow modulate them and silence them during infection process through genome editing tools, we can impart sheath blight tolerance. Also, we are trying to strengthen the rice defense against sheath blight disease to enhance sheath blight tolerance. And we have identified a very novel fungal eating bacterium, and I will try to emphasize how this bacterium can be helpful to manage sheath blight disease. Due to lack of time in today's talk, I will be only focusing on first and the fourth objectives. The second and third, maybe I will take another opportunity to share some of our work. So coming to the first objective, we observed that R. solanae, though it's a necrotrophic fungal pathogen, but it has initially a brief biotrophic phase where it establishes itself into the rice, and then subsequently it enters into the necrotrophic phase and kills rice to cause disease. And we did interaction transcriptome studies and RNA-seq analysis, wherein we sequence RNA of the infected rice, and then subsequently using computational analysis separate out the R. solanae gene and the rice genes and try to understand what all are the differentially regulated host and pathogen genes during infection process. So I'm not going to go into the detail of how we understand these genes. We have a couple of papers which will take you through, but the take-home message is that during RNA-seq analysis, we have identified important pathogenicity determinants of R. solanae that may be helpful for colonizing rice. However, one of the roadblocks in this field is there is no suitable method to study gene function in Rhizoctonia solanae. We cannot genetically transform the pathogen to study its function. So it has been a challenge in this field. So what we observed that R. solanae demonstrates similar mode of colonization in tomato and rice. Here I am showing you a disease infected tomato, and you can see the characteristic disease symptoms in tomato. And here I wish to emphasize that viral vector-based gene silencing approach is a good way to silence gene functions in tomato. And we have standardized a viral vector-based host gene silencing approach, wherein we express host RNA against fungal gene in the host and look for whether silencing of the fungal gene can reduce the pathogenesis. I am showing you now that, as I said, we had a couple of important pathogenicity determinants. One of them was RSCRZ1. It's a calcineurin responsive zinc finger transcription factor. And when we silence these genes using RNA Higgs approach, you can see the wild type plant has the characteristic symptoms. However, the silenced plant, we hardly see any symptoms. And this is the vector control plant. You can see the symptoms. So this shows that silencing of RSCRZ1 can compromise the pathogenesis of R. solanae. And here I am showing you indeed the expression of the fungal gene is compromised when we express the 6 construct in the tomato. Further, we observe that silencing of this gene compromises the growth of R. solani in planta. This is the wild type infected plant. You can see the infection fusions formation, and similarly in the vector control plant. However, in the RSCRZ1 silent plant, there is no infection fusion formation. The mycelium is only able to grow superficially into the rice plants. So this shows that RSCRZ1 is important for pathogenesis. I told you that it's a calcineurin responsive transcription factor. So we computationally predicted the target of RSCRZ1 and observed that many of them are upregulated during establishment phase. That means early phase of infection. These targets are also upregulated. And further, we used East1 hybrid assay to demonstrate that RSCRZ1 can indeed transactivate the selected target genes. And we silenced a couple of them to study their function. And here I am showing you that silencing of two of them, dioxygenase and substilicin-like protease. This too has been a target of RSCRZ1, also compromises the pathogenesis. So this work has been just published in MPMI. So with this, we demonstrated that the host-induced gene silencing approach and the dsRNA-based silencing approach can be effectively used to knock down pathogenicity associated genes of R. solani during infection process. And with this cartoon, I'm just trying to emphasize that in dsRNA approach, we spray the dsRNA targeting fungal genes, and in the Higgs approach, we make plants to express dsRNA against the fungal genes. And either we spray dsRNA or make plant to express dsRNA. This dsRNA is internalized into the fungal cells using vascular trafficking, and using fungal RNA machinery, the target genes get silenced. And if we end up hitting the important pathogenicity determinants, as in case of CRZ1, we can prevent the disease caused by the pathogen. So now we are trying to downregulate some of the pathogenicity genes alone and in combination to provide sheath blight disease tolerance in rice. So coming to the second approach, where we are trying to utilize an antifungal protein of a novel fungal eating bacteria for controlling sheath blight disease. You know, bacteria is a prokaryotic organism, it's a tiny organism. So while studying the rice rhizotonia interaction, we end up identifying a yellow colored bacteria from rice, which eventually we identify that Burkholderia gladuli strain NGJ1. So this Burkholderia, this is yellow in color here represented, and this is the Rhizotonia soli on a laboratory plate. You can see initially there is an antifungal zone. So this shows that during initial phase of interaction, it has antifungal zone. However, if we leave this plate for a couple of days longer, so around 7 dpi, this bacterium has now spread over the entire fungal biomass and has eaten away the fungal mycelia. Due to lack of time, I am not showing you data, but please take it from me that we have data to show that NGJ1 damages fungal mycelia and utilizes fungal biomass as source of energy. So this property, this fungal eating property is called mycophagy. So myco is a fungus and phagy is eating. My fungal eating property of the bacteria is mycophagy. But the mechanism of mycophagy has not been known. So we again did RNA-seq analysis, which revealed that type 3 secretion system, it's a needle-like secretion system. I have put a cartoon of it. So that through type 3 secretion system, bacteria delivers effectors into the host cells. So we found out that the type 3 secretion system effectors are upregulated during mycophagous interaction of NGJ1 with Rhizotonia solani. So there, I just want to emphasize that type 3 secretion system is a well-known virulence factor of the bacteria during its pathogenesis on plants as well as animals. But the role of type 3 secretion system during bacterial fungal interaction and especially mycophagy has not been known. So what we did, we mutated one of the core apparatus component, HRCC gene, to obtain a type 3 defective mutant of NGJ1, and we have shown that indeed the type 3 mutant is defective in inducing hypersensitive response, which wild type bacterium does in the host non-host plants. And when we subjected this type 3 mutant bacterium with R. solani, we observed that the mutant bacterium is unable to feed on fungi. Here I am showing you the top panel is fungus with wild type bacterium. The wild type bacterium is able to grow over fungi and it's eaten away the fungi. However, the type 3 mutant bacterium is unable to grow over the fungi and eat away the fungi. So this shows that type 3 secretion system is required for bacterial mycophagy. And as I said, that type 3 secretion system is a needle-like injection apparatus of the bacterium, and there are several effectors proteins being secreted into the host cell. So we try to understand what all are the effectors which is secreted through type 3 secretion system. So we had the genome sequence. We made the genome sequence of this bacterium and computationally predicted effectors protein of this. And one of them was a pro-fastal-like protein. And we demonstrated that indeed it is a bona fide type 6 T3SS effectors of NDJ1. We have data to show that the bacterium delivers this BG9562 protein into the fungal cells using type 3 secretion system. So we mutated this effector protein, BG9562 protein, and demonstrated that the mutation of this gene is compromising the bacterial ability to feed on fungi. This is the wild type bacterium has nicely eaten away the fungi. However, the mutant bacterium is unable to grow from the point of inoculation. You can see the yellow which reflect the bacteria, but rest of the plate is fungus. And when we complement the mutant, wherein we express the wild type copy of the gene, the complement behaves like the wild type. So this is a clear indication that this BG9562 protein, which is a pro-fastal-like protein, is required for mycophagy. So this work we published in Nature Communication, which is a prestigious journal in the field. So now we are trying to use this BG9562 protein for effective management of Rhizoctonia solani. So we observed that when we treat fungus with the purified protein, this is fungus treated with buffer. You can see the Arsenal is growing over the plate. However, when we treat Arsenal with the purified protein, the fungus fails to grow from the point of inoculation. You can see there is no fungal growth. And we observed that this protein is not only specific to Rhizoctonia solani, it has broad host range. So this protein has broad spectrum antifungal activity. We have got patents in some country and some other countries, the patent application is pending. So now we are trying to use this protein to control the sheath blight disease. Here I am showing you data. This is the Arsenal infected rice. You can see the disease lesions at the sheath. However, when we spread the protein solution, we hardly see the lesions. So this shows that protein spray is going to be effective in control of the disease. But now we are in collaboration with IIRR Hyderabad, especially Dr. Sundaram and Dr. Prakasham and Kannan Group, and IIRI New Delhi, where we are working with Professor A.K. Singh, Gopala Krishnan, and Vishnu Maya to make a formulation using this protein as an antifungal formulation and use it under the field condition to control sheath blight disease. And also we had this protein. So we made a transgenic rice overexpressing this protein. And here I am showing you two independent transgenic lines, which I am showing you L4 and L5. Both of them shows remarkable level of protection against sheath blight disease. This is the control plant, empty vector transform plant. You can see it's totally killed. However, both the transgenic plant is remarkably protected against the sheath blight disease. So this work we have done in pathogen inducible promoter. And as I said that this is a broad spectrum antifungal protein. So we are now assessing the tolerance of these transgenic lines against multiple fungal diseases, especially rice blast disease. So we feel that these transgenic lines will be tolerant to multiple fungal diseases. So in summary, I highlighted that the dsRNA-based approach and Higgs approach can be an effective alternative to silence pathogenicity-associated genes of Arsenal, and silencing of pathogenicity determinants can impart sheath blight disease tolerance in rice. I told you that there is no resistant genes, so here I wish to emphasize that understanding host pathogen interaction has given us an alternate tool to silence pathogenicity determinants to impart sheath blight tolerance. And also, we had identified a novel fungal eating bacterium. Fungus is a eukaryotic multicellular organism. Bacteria is a tiny organism. However, this bacterium can eat away the fungi. And we identified a novel antifungal protein, which we have named it as BG9562. And now we are exploring various means to utilize protein to control sheath blight disease in rice. So with this, I'm thankful to all my collaborators. I have wonderful collaboration with IIRR Hyderabad, IIRI New Delhi, ERI Philippines, CCMB Hyderabad, Nabi Mohali, RCB Faridabad, and IIR New Delhi, wherein we are working together under NIPDR flagship program. It's a DBT supported program to impart seed blight disease tolerance in rice. And also I'm thankful to my mentor, Dr. Ramesh Sonti, for guiding me, and all my lab members. I have a wonderful team of researchers who really work hard to make this work successful. So I'm thankful to all for making the work successful. With this, I complete.
Thank you, Gopalji. Excellent work you are doing. And I'm happy that you have also now Mycophagous bacteria, which can control sheath blight, which is one of the most dreaded disease in rice throughout the country now, particularly in central India. And I'm sure that the technology which you are now using it in collaboration with IRI and Hyderabad Institute, I think will find place in the farmers field. I'm sure that it will. So kindly continue the work. I congratulate you. Wonderful work.
Thank you, sir.
Now the last speaker of the lead speaker of this particular session, biotic stress management, is by Dr. J.S. Bentur. I saw that Jagdish Bentur is already around. Jagdish Bentur, you are welcome for this. He is currently working as a professor of biotechnology at Agriculture Biotech Foundation, Rajendra Nagar, Hyderabad. He is basically retired as a principal scientist entomology from the Indian Institute of Rice Research in Hyderabad, and he has long 4 decades of research experience in the field of host plant resistance against insect pests in rice. He has more than 100 research papers, and he is the recipient of Rafi Ahmed Kidwai Award, besides T.N. Ananthakrishnan Award, Bap Reddy Memorial Prize, Jana Reddy Award. He is a fellow of Royal Entomological Society, London, and fellow of National Academy of Agricultural Sciences. I remember when his application was there for NAS, I found Jagdish was always scoring in terms of the excellent publications that he has to his credit. So Jagdish,
thank you, sir.
Thank you very much for coming. And now the floor is open. We have, say, something like 20 minutes so that we get some time to at least have some questions. Thank you very much.
Sir, I'm audible now?
Yeah, you are.
Sir, I will take exactly 14 minutes, 50 seconds.
Excellent. Excellent.
Because this is a recorded presentation, I will play my the file. Please bear with me. Please bear with me.
Sir, you are sharing, sir?
Yeah.
Can you play Basavangoda?
Yes, yes, sir. I can.
He's sharing the presentation. Yeah.
Can you play my file?
Yes, sir. I will play, sir. I will.
Yeah, please.
Please play.
VLC player. Everybody, this is Bentur from Biotic Foundation, Hyderabad. I am going to talk on host plant resistance against insect pests. Screen.
Kindly take it on the screen.
Thank the organizers for giving me this opportunity. There is no screen, Basangoda.
Screen is not coming. Just check.
Hello, sir. Screen is not coming.
No.
Okay, then kindly make the loud voice. Kindly
sir,
enhance the voice.
I do not have control over the voice, sir, actually. That is pre-recorded voice.
Pre-recorded, okay.
Yes.
Jagdish?
Yes.
Please go ahead.
From Agri-Biotic
Now, sir?
Yeah. No screen.
No screen is coming.
What is this? Folder.
Can you put it on the screen? I'm not able to see the screen.
No, it's...
Yes, sir. Yes, sir. I'm sharing.
Okay. Everybody, this is Bentur from
No, sir.
No.
No. Double click it.
host plant resistance against insect pests? Bro,
Maybe 1 minute, sir.
if not, I can play.
Sir, can you share from your end, sir?
Yeah, I'll do it.
Yeah, you do it from yours.
Yeah. Can you see the screen now?
No.
No.
Share screen.
You just press the share screen.
Yes.
Ah.
Now we can see?
Yeah, it is coming now. Very good.
Okay. Okay, sir. Here it goes.
Yeah.
Oh, sorry.
Next.
Play.
Yes, Jagdish, it is coming.
Thank you, sir.
No
Sir,
audio, sir.
no audio, sir.
Can I go back and then present?
Yeah, the best way is you present now. You start from this slide, advantages and limitation of HPR.
Yes.
Yes, please.
Sound is coming?
No.
No.
Is sound okay?
No, sound is not good.
Not okay, sir. I think some problem is there. You can.
Can you hear me?
Yeah, your voice is okay, but it's not coming with the slide. Can you speak otherwise?
Yes, I think that is good.
Just by seeing the slides.
Can you hear me?
Yeah.
So, sir, I will try to cover it up as early as possible.
Okay, thank you.
One of the methods, what I have done is, this is a review of the work done. It's not my work alone.
Yeah.
In the host plant resistance, I split it into 3 phases. The first phase is before 1980. The middle phase is between 1980 to 2000. And the recent phase is after 2000 or so. So in the first phase, a lot of methodologies were developed and standardized. One of the achievement is development of a holidic diet for brown plant hopper, and in case of leaf folder, there is a meridic diet. Such diets are very important tools for investigating insect-plant interaction precisely. Just to make a comment here, the diet to rare yellow stem borer is still eluding the entomologists. Again, rearing rice gallmage on a medium free from the plant, I would consider as a millennium great challenge for the entomologists. Now I'm losing the track. Yeah. If the sound. So the early phases is the development and identification of resistant sources. I would recall that the initial attempts were made in case of gallmatch in India in 1950s. And it led to the identification of several breeding lines like CR55, CR56 from NRRI, and W1257 and W1263 from Warangal in collaboration with IIRR. Likewise, at the same stage, there were enough efforts made at IRI by MD Patek on striped stem borer and by Heinrichs on plant hoppers. So all these sources of resistance have been scattered. There is no single place where we can look or get the information by the researchers. And the second thing is that it is still difficult to get an authentic seed source of the reported sources. In the first phase, however, some of the problems encountered could not be resolved, like high level of resistance against yellow stem borer, or lack of precise knowledge of genetics of resistance leading to incomplete transfer of resistance in case of BPH, and prevalence of biotypes of gallmatch, and the durability of resistance in case of BPH and the gallmatch. So in the middle phase, where the genetics, genes, and marker information was made available, a lot of advancement was made in this phase. Let me go through this one. The first impact was on genetic identification of major resistance genes. There are 40 major genes reported against brown plant hopper, thanks to the main leading work of Dr. G.S. Kush and his associates. And in case of white-backed plant hopper and gallmatch, at least 12 genes have been reported in both the cases, and several genes in case of green leaf hoppers. But with the publication of Alam and Cohen, the approach to insect resistance has been transferred or been diverted from major genes to QTLs. There are hundreds of QTLs being reported. It is being influenced by the parents of the mapping population, phenotyping methods, environment, and pest population. But the utility of several of these minor QTLs in enhancement of insect resistance is yet to be clearly demonstrated. Again, the markers have come in a big way, and this development of markers has helped to tag and map resistance genes. In case of gallmatch, 10 out of the 12 genes have been mapped on 4 to 5 chromosomes. In case of brown plant hopper, 30 of the 40 genes have been mapped. Significantly, 28 of these genes are in 5 clusters on 4 chromosomes. The significance of these clusters are discussed in some of these review papers. But more important, the direct spin-off of this tagging and mapping is availability of the flanking markers, which can be used for introgression of the gene through marker-assisted selection. Here I list some significant publications which have used markers to pyramid 2 or more genes, not only insect resistant gene, but also other diseases like the blast, BLB, and also in case of brown plant hopper. So this is the second flush of products where these are available to the farmers. Another issue of gallmatch biotypes has been very successfully resolved during this second phase by characterization of the gallmatch biotypes, showing spatial and temporal distribution of the gallmatch biotypes and biotype composition of the gallmatch populations in India. However, gene for gene relationship in this case is yet to be established, and this is a challenge for the next generation researchers. In case of brown plant hopper biotypes, it is still a contentious issue. Somebody says there are biotypes, somebody says there are no biotypes. However, elegant work of Horgan, who is our the lead speaker today, he has demonstrated the virulence of different populations in South and Southeast Asia against the rice varieties, and also the durability of these genes. Another significant development is transgenic rice. Though transgenic rice does not represent the natural host plant resistance, but resistance is impregnated into the host plant. There are more than a dozen genes that have been used for developing resistant transgenic rice against lepidopteran pests and sucking pests. Unfortunately, these transgenic rice are not available to farmers for cultivation. More significantly, now transgenesis has become an indispensable tool for functional identification of resistant genes. Now, the latest phase, or the third phase, where I'm struggling to keep pace with, there have been several developments like sequencing of rice genome, resequencing of 3K genomes, improved markers, cloning of R genes, and several related fields. So one of the recent development in marker technology has improved genotyping precision, like different SNP genotyping or genotyping by sequencing, superior haplotype identification, or genome-wide association studies. I lost track of my screen.
Sir, actually screen was paused actually, sir.
Oh. Yeah, the other development is the genome sequencing of the insects, like the brown plant hopper, the small plant hopper, and the white-backed plant hopper. This information is giving us which are the key genes in the insects that can be targeted. I hope I will go to next slide. Yeah, like also metagenome. Metagenome is the genome of mitochondria. Mitochondrial genome sequencing also has been done, which gives us the idea or the tools to study the populations as well as develop the markers for biotypes. In addition to mitogenome, metagenome is another focus of study where the associated endosymbionts in the pest, the role they play in conferring the survival of the insects by synthesizing the essential amino acids to confer virulence. Developments or the application of omic tools has elaborated the pathway of defense. We now know that the defense mechanism against chewing insect mediated by JA pathway is significantly different from the resistance against sucking pests, which is mediated by salicylic acid pathway. Likewise, our studies on gallmage has highlighted the difference between the resistance associated with hypersensitive reaction and resistance independent of hypersensitive reaction. The recent developments in RNAi-mediated insect gene silencing is targeting the insect genes through the rice plants. And finally, the genome editing is targeting the what you call susceptibility genes, which Dr. Jha has just highlighted. So all these the tools are now available, and some of them are very powerful tools. Time will tell how powerful these tools are. But the question whether man can beat the insect has already been answered. The answer is a definitive no. Thank you very much for your patient listening.
Thank you, Dr. Jagadish.
Sir.
I think we completed well in time. There are, if there are any few questions, because we have quite a good number of participants who are now attending this. I would just like to summarize for their benefit if they have any questions. You see, the first lecture was a keynote lecture by Finbar, who gave a very detailed account of eco-friendly management of rice pests. What is a lot of experience in Australia and in Philippines, which he has carried out. And the significance he has concluded that instead of IPM now, we should really look into what we call it as integrated crop management. And there he says that instead of there may not be any necessity of what we call it as a curative action for pesticide. If we make a proper balance, biodiversity, predator, parasite, and they are all getting actually eroded mainly because of the misuse of pesticides in the rice ecosystem. I think it was one of the very fine lectures that he very well depth studied lecture. Mohan Kumar gave an excellent exposition of stored grain pest. I think there may be a few questions on this, but he has uh, specifically said what are the new techniques, mapping of the level of phosphine resistance, and assess the structure of grain storage pests. That is very important. And he also developed novel microsatellite markers developed in rice for rice weevil using the next generation sequencing technologies. So I think Mohan Kumar did justice to what we were expected to do in rice, because there is a huge problem of rice storage today because of the heavy production that we are crossing more than 110 million tons of rice. So I think there are several issues. And as he said, that the new silos which are coming, maybe we'll have to have specific silos for rice and other things. The third lecture was specifically Gopalji Jha's, his own set of students and wonderful work which he has carried out. And what he has said is really very good, that the sheath blight has been a very serious problem. And the management of sheath blight using the transgenic approach, and also using what we call it as sort of the bacteria attacking fungi. I think mycophage fungi, bacteria, something quite a good unit, and this can be really converted into lot of. And as I told, his collaboration with others is very useful. The last lecture, I think there was some Jagdish. I'm sorry, because although Jagdish has done a wonderful work in host plant resistance in rice, particularly in the insect pest. But I think because there was some mismatch in the slide position, we many of us could not hear about it. But let me tell you that if you go to his some of the papers, which I happen to have read when he applied for NAS, it's a really wonderful work which he has done in these areas, and which needs further attention, particularly RNAi, and now what we call it as genome editing technologies, which will be very useful in rice. And I'm certainly sure that the workers will be benefited. So this overall session of our biotic stress management, where I had the collaboration with Dr. Chakravarti, we had Dr. Rath Mandal as our partners in reporter. I hope they have made the notes of it. I would, I think, take up one or two questions if there are any specific questions, so that then we can conclude. Dr. Mandal, is there any questions in your question box?
So just let me check, sir.
Yeah.
So only one question was there, but that was a different that he wanted to know about. That is there in talk any related to microbe-mediated biotic stress tolerance? Microbe-mediated biotic stress tolerance in rice.
This was for Dr. Jha is around?
I'm around, sir.
Microbe-mediated biotic stress tolerance.
Yes, so my talk mostly covered that how a microbes can be helpful to control disease. And I just wanted to take this opportunity to highlight that there are many useful microbes in the plants. So if we can, and I just showed you an example how a rice-associated bacteria can be so helpful to manage the disease. So there are many such bacteria, there are many such microbes associated with plants. And if we can in-depth characterize them, it will be a wonderful opportunity for the researchers and to manage the disease. So that will be my take-home message to encourage the researcher in this field.
Dr. Gopala,
yes,
there
sir.
may be, I think, smaller than bacteria organisms which may be mycoparasitic in the system.
Yes,
I
sir.
think they need to be more investigated. But I
So there
doubt
are
very
many,
much.
yes, sir, there are many kinds of microbes which is associated with the plants.
Yeah,
so nowadays the concept is the microbiome modulated function
Biota,
in
yeah.
plants. So if we can modulate microbiome and make to have more beneficial microbes than the harmful microbes, they can be able to systematically affect the plant physiology and help us to have better crop.
Very good.
So,
Thank you very much.
Yes, sir. Thank you.
So, sir,
we are got in time. Yes.
Questions,
Any
questions, yes,
yeah.
sir. Another question is to Dr. Bentur that will RNI also work for BPH and WPH? Do a variety will practically possible?
Jagdish.
Bentur sir, Jagdish sir.
Bentur, your mic is kindly make it unmute.
You have to unmute, sir. Again, I read the questions. Will RNI also work for BPH and WPH? Do a variety will be practically possible?
If you can hear me, can hear me, can you hear me? Okay, sir. Yeah, there are many genes which are being targeted by RNAi mediated from the brown plant hopper. We have a recent review that is coming up with Sundaram and Mangruthia. We have reviewed this, the possibilities of using different genes against plant hoppers. Yes, answer is yes.
Thank you, thank you. Yeah. So, Dr. Mandal, I think we conclude this session. It is exactly now almost 4:30. I think we could manage the time very well. I really congratulate all the speakers who have done a wonderful job for this session. And there will be now oral presentations in biotic stresses subsequently. So thank you very much, Mandal. I hand over the mic to you now.
Yes. At the outset, just I would like to profusely thank our CD Mai sir. I think he's now the president, South Asia Biotechnology Center, and he's always a guiding inspirations for all of us in different platforms. And also, I would like to profusely thank Dr. P.K. Chakraborty, who is a member ASRB, for his gracious presence of this co-chairing the sessions. And we are really thankful, sir, both of you, for kindly chairing these sessions, and despite of your busy schedule and valuable time. So I also thank to all presenters, and I think it was a thoroughly insights about the insect pest and insect pathogens interactions. And I'd like to especially thanks Gopalji Jha, who has taken my invitations, and because we have been working in long time in associations. And also to others also, it is a very good insight from all of you, sir. And also thanks to our convener, Dr. P.C. Rath, sir, and also to nicely convene the sessions, and also to rapporteurs who has taken a note which will be delivered in the plenary sessions. And thanks to all participants, and particularly also thanks to the technical host who has helped us in convening these sessions. Thank you very much.
Thank you, Basana Gowda.
Yes, sir. Thank you, sir. Sir, we have one announcement to make.
Yeah.
After this session, we have a session on oral presentation, which was supposed to start at 4:30. We are already 5 minutes late. I request everyone to join that session using a different link, which was already sent to you. Okay. You kindly see the program. So there is this link will expire in a few minutes. So we have already given that link in the program. So kindly join that oral presentation session using that link. Thank you. Thank you very much.
Thank you.
Thank you, everyone.
Thank you.
Thank you, Dr. Finbar. Okay, bye.
Nice to see you again. Goodbye.
Goodbye. Okay, thank you.
Sir, இந்த வாரம் அந்த டிபிடி அக்ரி பேட்டர்லாம் மாத்திட்டாங்க.
அப்பதான் ஒரு சந்தேக கலைகள் பண்ணிட்டாங்க. அவங்க வேற செக்ஷன்ல நினைச்சுப்பாங்க.
ஹெச்ஆர்டி சார், எம்எஸ்சி பிஹெச்டி எஜுகேஷன்.
வேற எம்கே மோடின்னு போட்டிருக்கு. அவர் ஏற்கனவே இருந்தவர், இதுல இருந்தாரு. ஒரு ஆமா எங்களுக்கு ஒரு செக்ஷன்ல இந்த பயோ ரிசோர்சஸ்ல இருந்தாங்க. நல்ல டைப்.
அப்புறம் சார் இந்த நம்ம இன்ட்ரெஸ்ட் ஏண்ட வந்து அந்த பாரத் கோர்ஸ்ல ப்ரொசீடிங் கொடுத்துட்டாங்க. நம்ம அக்கவுண்ட்ல வந்து நெட் பேங்கிங் கிடையாது. நம்ம டிபார்ட்மெண்ட் அக்கவுண்ட் இருக்குது பாத்தீங்களா?
ஆமா,
அது எதுவுமே நெட் பேங்கிங் கிடையாது சார்.
நெட் பேங்கிங் வேண்டாம். அவங்க கிட்ட ஒரு லெட்டர் எழுதி கொடுத்து
மோகன்குமார் சார், நீங்க வந்து மியூட் பண்ணாம பேசிட்டு இருக்கீங்க. மோகன்குமார் சார், நீங்க மியூட் பண்ணாம பேசிட்டு நல்லா இருக்கீங்களா?
சாரி மேடம். நல்லா இருக்கேன் மேடம். நல்லா இருக்கேன்.
ஓகே.